Hello, this is the hematopoietic and normal blood cell hematology tutorial presented by the SchrDbb with reference Scholar Atlas of Hematology by Harald Thelm. So here you're going to visualize hematopoietic cells and normal blood cells hematology. So on this blood section, what you visualize is actually early erythropoiesis. So this is a diagram of early erythropoiesis. So in the diagram of early erythropoiesis, we have this diagram A, which is the earliest recognizable red cell precursors. Is it clear? The earlier recognizable red cell precursors are going to be called the pro-erythroblasts. Is it clear? And the pro-erythroblasts are shown in the diagram 1. So these are the pro-erythroblasts here, and they are arranged with nuclear chromatin inside. You see, there's a very large nucleus here, so it tells you that it is the earliest form of the erythroblast, which is the pro-erythroblast. Is it clear? So below are two orthochromic. So now so these two here are two orthochromic erythroblasts. Is it clear? So after the pro-erythroblast, you have this orthochromic erythroblast, which comes later. And then on this side here, we have the metamyelocytes, and the metamyelocytes are involved in the white blood cell hematopoiesis. It's not in the red blood cell hematopoiesis. So you see, this metamyelocyte has a small U shape. We are not going to speak about that in red blood cell hematopoiesis. So in red blood cell hematopoiesis, you start with pro-erythroblast, then we have orthochromatic erythroblast. Is it clear? So when I say orthochromatic, it means that it only has one color. So the next one here, we have in diagram B, we also have the pro-erythroblast. So this is another pro-erythroblast that is shown here in this diagram. Is it clear? So and we also have a myeloblast here, but this one is not involved in red blood cell as a red blood cell precursor. Is it clear? Now the next one here at this level, we have also here pro-erythroblast. Here at this level here, we have different diagrams involved. So at this point, we have a pro-myelocyte here. Here we have here we have pro-myelocyte at this level, and then here we have metamyelocytes, which are not involved in red blood cell production. So now the next diagram here is going to be involved now with the dense. So this figure is still on the erythropoiesis, cells involved in erythropoiesis. So for the cells involved in erythropoiesis, we start with this first part here. So these are the nucleated erythrocyte precursors. So we see that in the nucleated erythrocyte precursors, this is in diagram A, we have basophilic erythroblast. So we started, the first one started with pro-erythroblast. The second one then was the orthochromic erythroblast. Then we have now what is called the basophilic erythroblast with condensed chromatin structure. Then after the basophilic erythroblast, we have now the polychromic erythroblast. Is it clear? The polychromic erythroblasts are shown here, and the polychromic erythroblasts have different colors at the level of the cytoplasm. There you need to know that the explanation for this change in colors is just because at first in pro-erythroblasts you only have a very large nucleus which exists as euchromatin. Is it clear? Then later on, the nucleus produces ribosomes. Is it clear? The more the nucleus is going to produce ribosomes, and the more hemoglobin is going to be produced, the cytoplasm is going to change pinkish. So at first it's going to be called pro-erythroblast. Then you have orthochromic erythroblast, which is only bluish because there is no hemoglobin produced. Then you have basophilic erythroblast, which is produced because because you're going to have only the nucleus. Then you have polychromic erythroblast is going to be produced when you have polychromic erythroblast is going to be produced when you have the color of the cytoplasm is both pinkish and bluish, as you can see here. So the first is pro-eritoblast, the second is basophilic eritoblast. Sorry, it's not autochromic, it's basophilic eritoblast. The third is polychromic eritoblast, and then the fourth is autochromic eritoblast when it is becoming pinkish. Cytoplasm is becoming more pinkish, as we've seen in the last diagram. So now this is another diagram here. Diagram B, you see erythropoiesis is being involved here in the bone marrow, and then it is organized around this cell here. This cell is a macrophage. You see how it has a U-shaped nucleus, and then it has a very large cytoplasm. It's a macrophage, and then this macrophage has erythroblast close to it here. These are the different erythroblasts. This erythroblast can be said to be autochromic erythroblast. Is it clear? Now the next one, this diagram, this is now diagram D. Diagram D now is involved with to show similar lymphocytes. So you have lymphocyte and you have erythroblast. You see that lymphocyte and erythroblasts are quite similar, though erythroblasts have a greater cytoplasm compared to the lymphocyte, just to distinguish. So this is actually the how erythropoiesis occur. To know the steps of erythropoiesis, the first thing that occurs is the first thing that occurs is the pro-erythroblast. Then you have the erythroblast. Then we have inside the different erythroblasts. The first erythroblast that is basophilic erythroblast, because most of the cytoplasm is going to exist with a base. Is it clear? Then the next one, we are going to have polychromic erythroblast. Then you're going to have autochromic erythroblast. After autochromic erythroblast, now we have reticulocytes, and then lastly you're going to have red blood cells. So that is how that is going to be produced. Now this step here is going to be granulocyte precursors. So we are finished already with erythropoiesis. Then we are going now into granulopoiesis, the production of granulocytes. So in the production of granulocytes, you have granulocyte precursors. So in diagram A, you are going to have mature precursors in the granulopoiesis. But before we start with mature precursors, you need to know that what all the precursors from of granulocytes start from a myeloblast. Is it clear? And you can identify myeloblasts here in this diagram. So this diagram, you have a myeloblast here. All precursors are starting from a large myeloblast. So in diagram B, you still see this as a myeloblast, and this is already a neutrophil which has completely been formed. Is it clear? In diagram C, so in diagram C, what do you have? So in this diagram, you can identify the first one, which is a myeloblast here, and then the second one is a promyeloblast. A promyeloblast is what occurs before is a precursor of a myeloblast. So in this other diagram also, we still have, now we have myelocytes. After myeloblast production, we have myelocytes, and after myelocytes, we have the second one is metamyelocytes. After metamyelocytes, now this one here is the third one is a polychromic erytoblast. Polychromic erytoblast is not involved with white blood cell production. So we have myelocytes and metamyelocytes involved in the white blood cell production, and then this one is so the third one is metamyelocytes. So the first one we said myelocytes, so and all that promyelocytes, myelocytes, and metamyelocytes, and this one is polychromic erytoblast, not involved in white blood cell production. So in general, the white blood cell for the granulopoiesis, you need to know that there are 3 main granulocytes, and all of them is stimulated by clonal stimulating factor. The 3 main granulocytes are neutrophils, eosinophils, and basophils. Is it clear? Now each of them are going to have their specific name just before the different precursors I'm going to call. So the first precursor that we are going to see is a promyeloblast. After the promyeloblast, as you can see here, we have a myeloblast, as you can see here. After the myeloblast, the next one you have the promyelocyte. After the promyelocyte, you have the myelocyte, the metamyelocyte, and then we have the myelocyte. Is it clear? You have to know that what the metamyelocytes have a U-shaped diagram. So their shape, the shape of their nucleus is U-shaped. That's a specific characteristic of a bean shape. the specific characteristic of a metamyelocyte. Is it clear? So those are the different things that I wanted to speak about here. Now, on this diagram here, we are going to visualize myelocytes and metamyelocytes. So this is the specific diagram of each of them. So the early myelocytes can be seen in diagram A. So you can visualize the early myelocytes here. We have chromatin structure is denser than that of the promyelocytes. I can see you have in early myelocytes the chromatin is denser than in the case of a promyelocyte. Why? Because the promyelocytes, you need to know that what the earlier you are in hematopoiesis, the more your chromatin is going to exist as euchromatin. Is it clear? And the more you are at the end of hematopoiesis, the more your chromatin is going to exist as heterochromatin or in condensed form. Is it clear? That's why you have a promyelocyte is going to have this one here is a myelocyte. You already see that there's a dark bluish portion which corresponds to the heterochromatin or the condensed form. Is it clear? Now, in this activated, this now is activated myelocyte here in the cytoplasm. So activated myelocyte here in diagram B. So the next one is diagram C. We have typical myelocytes also. So you see heterochromatin that you can visualize in this myelocyte, condensed heterochromatin. This is already a neutrophil has completely been produced. And this diagram D show a metamyelocyte is distinguished from the myelocyte by the lobe formation. So here you have a bean shape of the nucleus. Is it clear? So the first one is the promyelocyte. The promyelocyte is going to have a nucleus which is completely clear because contain of euchromatin, while the myelocytes now, the nucleus is going to be a bit darkish because it is made up of more of condensed chromatin. And then the last one is metamyelocyte is going to have a bean shaped nucleus. Now, the next thing you're going to visualize is the neutrophils. You are going to visualize the band cells and all that. So here, this first diagram, this diagram A, you have transition form between a metamyelocyte and a band cell. So immediately after the metamyelocyte, you have a band cell. You see, it is more of a bean shape. So the bean, that curvature increases more in case of a band cell. So to leave from a metamyelocyte to band cell, you have an increase in curvature. Is it clear? So in diagram B, you have copious granulation in band cells. So you see, in band cells, you have more granulation are produced. Why? Because you need to know that what the more you are at the end of hematopoiesis, the more you are condensing your nucleus, and the more you are producing granules or substances in your cytoplasm that will help in the function of that particular cell. Is it clear? That's why the band cell is going to have more copious granulation inside the cytoplasm. Is it clear? So we have toxic granulation. Is it clear? So in diagram 2, you can visualize the Dole bodies with the arrow. So we have the Dole bodies which can be seen with the arrow. Is it clear? So in diagram C now, you have band cell here, you have another band cell here. So these are the two band cells that you can see with the Dole bodies. You have Dole bodies inside like that. Is it clear? Now in diagram D, you can visualize band cells still which are trying to aggregate with each other. And then in diagram E, you're going to see segmented neutrophilic granulocytes. So these are when they are already segmented, the curvature has increased more such that they are segmented, and the segmentation is irregular. Is it clear? In diagram F, we are going to have segmented neutrophil granulocyte after peroxidase reaction. So when there is already peroxidase reaction, it becomes like this on light microscopy with hematozidine and eosine stain. Is it clear? And the last one here is segmented neutrophil granulation when there is alkaline leukocyte phosphatase staining. So this is another stain. If you don't use hematoxylin stain, that's going to stain it normally like that. This is with peroxidase stain. Is it clear? This is hematoxylin and eosin stain, and this is alkaline leukocyte phosphatase staining. Now the next diagram here is going to show you variation in segmented neutrophils. Is it clear? So diagram A, we have reactive state with toxic granulation. So this is at the reactive state with toxic granulation, more visibly expressed on this one. You see, this is going to be more toxic. So here see the granulation, there are more granulations compared to this other one. Is it clear? Now in diagram B, you have sepsis with toxic granulation. You see the granulations are trying to be be moving outward. So you have vesicles formation such that the granulation can be released. It shows that this patient is going to have a severe infection. Is it clear? Now we also have that in diagram C, we have what is called the Pseudoburger cells looking like sunglasses. So you see, you have sunglasses like this, and these cells are called Pseudoburger cells. Is it clear? So they are toxic, or they are myelodysplastic cells of neutrophils. So this type of cells are still neutrophil, which are polysegmented, and they are toxic and or they are myelodysplastic. So this diagram is just to show you some pathology condition that can be related with respect to hematology and neutrophils. So this, you see that there are many granulation, and the granulation are very rough. As I've said, this one is toxic granulation. It shows that this patient is having a sepsis. Also here you see there's also toxic granulation, and the other granulation is also put in vesicles, and they're even released. So it shows that this patient already have sepsis, severe infection. In this case, you have sunglass appearance. That is Pseudoburger cells. Pseudoburger cells just tell you that you have a myelodysplasia or you have toxicity, but toxicity is more of granulation, while Pseudoburger cells is more of myelodysplasia. Means that there's a certain dysplastic condition, like a virus infection, that may change the cells to tumoral cells. Is it clear? Now in diagram D, we have the dough-like basophil inclusion. So these are the basophilic inclusion here, which are dough-like, those dough-like basophilic inclusions. So those dough-like basophilic inclusion without the toxic granulation can be seen here. Is it clear? Now the next one, in this next diagram, now we are going to visualize still neutrophilic clinical correlations, the clinical correlation of neutrophils. Here you're going to see hypersegmented neutrophilic granules. Is it clear? It has 6 or more segments. So here, so it can be seen in megaloblastic anemia. This hypersegmented neutrophil here can be visualized in megaloblastic anemia. Now this one has clinical correlation. So diagram F has a clinical correlation in the fact that it is the drumstick. So this is drums. You can visualize in this diagram, you can visualize drumstick here with arrow 1, and you have appendix in the nucleus, and this one is associated with X chromosome. So it's an X chromosome disorder with drumstick. So we have a segmented nucleus with a drumstick here, usually associated with X chromosome disease, X chromosomal disease. Now diagram G shows you a very large granulocyte from the blood sample taken after chemotherapy. So granulocyte is going to be very large, and you see the segmentation is irregular. So this is in case of a patient taking chemotherapy. Is it clear? And now this one, diagram H, is a segmented neutrophil granulocyte during degradation after seen as artifact prolonged storage. So here is when the neutrophil want to degrade after doing apoptosis. When apoptosis wants to occur, the segmentation become like that in neutrophil. Is it clear? So those are the different cases of neutrophils. Now we go now to eosinophilic granulocytes and basophilic granulocytes. Now eosinophilic and basophilic granulocytes can be visualized in this diagram. So diagram A to diagram C, you can visualize eosinophilic granulocytes. See, these are eosinophilic granulocytes, and you need to know that what the specific the difference between the multilobar nucleus of eosinophil and the multilobar nucleus of neutrophils is that multilobar nucleus of eosinophil is more oval in shape, while multilobar nucleus of neutrophil is more irregular in shape. So that's the difference. So this one is eosinophil, this one is eosinophil. Is more irregular in shape. And then the granulation of the neutrophil is more clear or bluish in color. The granulations of neutrophil is more clear or bluish in color inside the cytoplasm, while the granulation of the eosinophil is more pinkish in color. So the granules are going to mostly contain, in eosinophil they contain mostly acids, while the granules in neutrophils contain mostly bases. Is it clear? So now diagram D, so here is the diagram D. So these are the different orange stained granules which are found here. So in diagram D is a neutrophilic segment which is not having a round. So here you see, can visualize a neutrophil here, and then it's polysegmented. It does not, the segments are not oval as you can see in this case here for eosinophils. Now diagram B is a basophil and with basophilic granulation. The basophilic granulation, you need to know that what the basophilic granulation are very bluish as compared to in contrary to that of eosinophilic granulation, which are pinkish. Basophilic granulation are bluish, and it may even cover the nucleus, which also is bluish. So that's how you can visualize a basophil, just having bluish cytoplasm and bluish nucleus, which is overshaded by the cytoplasm. Is it clear? The next diagram now that we're going to visualize are the monocytes. So in the monocytes, so this diagram we have range in appearance of typical monocytes with lobed nucleus. So you can see gray-blue stained cytoplasm. So these are gray-blue stained cytoplasm because the cytoplasm contains lysozyme, and that lysozyme is also basic. So it is this. That's why it's going to make the cytoplasm to be bluish. Is it clear? And then you also have a nucleus here that is going to show that you have a monocyte. Is it clear? So from diagram A to diagram C, you can visualize monocytes. They are very large with gray bluish cytoplasm, and then they are not polysegmented. They are larger than neutrophils. That's the first thing that you have to know. And when they are larger than neutrophils, they are not polysegmented. So you see that nucleus is only like this. So it's like a U-shaped nucleus, just like that, and then the cytoplasm is bluish in color. Is it clear? So gray bluish in color. Now diagram D show phagocytotic monocytes with plasma vacuoles. So these are the different vacuoles here where when he has already done phagocytosis. Diagram E now is monocyte that shows, so you have monocytes here on the left, shows what? It shows azerophilic granulations. So when you are being told of, so we have azerophilic granulation, is just saying that it is bluish. Is it clear? So when you are told that something is azerophilic, it is bluish, meaning that it is absorbing azure. Is it clear? So when you are azerophilic, you are bluish, and when you are eosinophilic, you are absorbing eosine and you are pinkish. Is it clear? Azerophilic is just this, is similar to basophilic, so both of them are bluish. Is it clear? Just that basophilic is that you are going to, so azerophilic is that you are going to absorb azure, that is a stain that's going to make you to be bluish, and then basophilic is that you are going to absorb methyl blue, that is also going to make you to be bluish. Is it clear? So those are the different things that you are going to visualize. So these are the monocytes. Still monocytes here in diagram F, you see have monocytes with nucleus resembling that of a band nucleus. You see that the band nucleus that you have seen at the level of the precursors of the neutrophils, that had a U shape, a over curved U shaped. This is also a monocyte. This is a monocyte resembling that band nucleus. So in diagram G now, you have a monocyte that has phagosized with vacuoles inside, and this is diagram H with esterase staining. So the staining method that has been used here is esterase, is not the same one that we know. Now the next one is lymphocytes. So in lymphocytes now, we need to know that from diagram A to diagram C, these are so range of lymphocytes. Some of them are adjacent. So here these are different lymphocytes you can visualize, and these are the neutrophils which are adjacent to the lymphocyte. Is it clear? Now diagram D shows in units that there are some lymphocytes from neonate that show irregularity in nuclei. So in units you can have irregular nuclei like here. In diagram E you can visualize in diagram E you can visualize also this. Diagram G you can see a smear taken after infection that may occur. So you see, after an infection, the lymphocyte, you see this lymphocyte here has a very large nucleus because it is a naive cell, a cell that is not actually already it has not already been adapted to a particular antigen. So this one is a naive cell, and a naive cell contains a very big nucleus. Is it clear? Now in this case, the naive cell has been exposed to an antigen, and you see the cytoplasm of the naive cell is much more bigger, as the cytoplasm of this type of cell is much more bigger than the naive cell, and these cells are called plasma cells. And plasma cells are the one that are, or memory cells. So memory cells, plasma cells are the one that are going to respond to the next interaction with an antigen. Is it clear? When this particular cell is a plasma cell. And to visualize plasma cell, you need to know that there is an o'clock appearance of the nucleus. Is it clear? When in this case, you can see it's in like memory cells and all that. Now in this case, we have now megakaryocytes and thrombocytes. Is it clear? So these are megakaryocytes and thrombocytes. So at this first diagram here, you're going to visualize megakaryocytes. They are very large. So in the eritopoiesis of megakaryocytes, start from megakaryoblast, then it goes to megakaryocytes, then it goes to the thrombocytes due to the fragmentation of the cytoplasm of the megakaryocyte. So here in diagram A, we have megakaryocytes in the bone marrow smear. Is it clear? Then so that is diagram A. Number diagram B, we have now normal thrombocyte. Thrombocyte you can visualize here. Is it clear? You can see thrombocyte. And then in diagram C, you can also visualize different thrombocytes here. You can visualize different thrombocytes. So thrombocytes are the smallest blood cells inside the blood cells. Is it clear? And they don't contain any cytoplasm. They don't contain any nucleosoid, but they contain only cytoplasm with different granulation. And you see that the granulation is absorbing azure or absorbing methylene blue. That is why they can be said to be azurophilic or basophilic, because they are bluish. Is it clear? So from here we are finished with the first part of the tutorials and continue the next part. Thanks for your kind attention. Don't forget to like and subscribe for our channel. Thank you.